When it comes to making medicine, nothing matters more than patient safety. And a huge piece of that puzzle is cleaning. But we're not talking about just wiping things down. We're talking about a level of clean that has to be scientifically proven, which all comes down to a really high-stakes question.
Think about it. How can you be absolutely certain that the equipment that just made, say, a potent painkiller is safe to make a children's vitamin next? Even a microscopic amount of leftover residue could be a disaster. You can't just guess or hope for the best. You need a number, a hard, scientifically proven limit.
So how in the world do we get to that number? Well, that's exactly what we're going to break down. First, we'll look at the core problem of contamination. Then we'll open up the scientific toolkit used to solve it. We'll get into the actual calculations, see how the final, critical decision is made, and then we'll see how all this theory gets put into practice on the factory floor.
You know, you could look at a massive, gleaming stainless steel tank and think, wow, that thing is spotless. But in the world of pharmaceuticals, what you can see with your eyes just isn't the standard. Invisible particles, things you'd never notice, can still be there. And that's why looking cleaned just doesn't cut it.
And that's where this concept, MACO, comes into play. It stands for Maximum Allowable Carryover. This isn't just some guideline. It's a hard, calculated number. It literally defines the maximum amount of gunk from product A that is scientifically proven to be safe if it ends up in product B. MACO is our line in the sand. It's our safety threshold.
Okay, so how do we find this magic number? Well, scientists don't just pull one method out of a hat. They have a whole toolkit of different approaches. And you can think of them like different lenses, each giving you a slightly different but super important view on the same safety problem.
Inside our toolkit, we've got three main instruments. There's the 10 parts per million criterion, the therapeutic dose criterion, and the health-based exposure limit criterion, which everyone just calls HBEL. So before we dive into how they each work, let's get a quick side-by-side look at them.
What's really interesting here is you can see the different philosophies at play. The 10 ppm rule is all about being simple and straightforward. The therapeutic dose method zooms in on the actual strength, the potency of the drug itself. And HBUL, that's the most comprehensive of all, looking at the entire toxicological picture to assess the total risk to a patient.
All right, let's get into the mechanics of it. This is where we see how those different ways of thinking actually turn into real numbers. And don't worry, this isn't going to be a big math test. We're going to focus on the logic behind the formulas. First up, we have the 10 ppm rule. This one's a classic, a real old-school industry standard. It's a general catch-all limit. It basically says, I don't care what the last drug was, you can't have more than 10 parts of it for every million parts of the new drug. It's simple, it's easy to understand, and it's been used for a long time.
And the logic is really straightforward. You take that concentration, 10 parts out of a million, and you just multiply it by the batch size of the next product you're going to make. So yeah, a bigger batch might technically allow for a little more residue in total milligrams, but the concentration always stays at or below that safe 10 ppm level.
Now let's move on to our next tool, which gets a lot more specific. The therapeutic dose criterion says, hey, a general limit is fine, but let's look at the actual power of the drug we're cleaning up. The whole point here is to make sure that any residue is such a tiny, tiny fraction of a single dose, like 1/1000th, that there's no way it could have any medical effect on a person.
So instead of a generic number like 10 ppm, this calculation is actually based on the specific dosage of the previous drug. And that's its huge advantage. It customizes the safety limit to the actual risk. A really potent drug is going to get a much, much stricter limit than a less potent one. It just makes sense.
And that brings us to the gold standard, the health-based exposure limit, or HBEL. This is absolutely the most modern, most scientifically rigorous way to do it. It goes way beyond just the drug's intended effect. It's a deep dive into all the toxicological data out there, animal studies, human trials, everything, to find a level of exposure that's considered safe for a person over their entire lifetime. The formula here starts with this really critical number called the PDE, the permitted daily exposure. That number comes directly from all that deep toxicological research we just talked about. Then, you use that PDE along with some batch and dosage info to calculate a MACO that is tied directly to a really comprehensive understanding of long-term patient safety.
So now we have three different tools, and they could give us three different answers. So which one do you pick? Well, the answer comes from a very simple and very powerful idea. You always plan for the worst-case scenario.
In the real world, you don't just pick your favorite method, you have to run the numbers for all of them. And your goal is simple. Find the lowest possible MACO value.
This is all about using the most conservative, most strict limit possible. Let's see how that plays out.
So let's imagine we're cleaning some equipment. We run the simple 10 ppm calculation, and it tells us our MACO is 1,000 milligrams. Okay, that's our first number.
But then we run the therapeutic dose calculation. And because the drug we're cleaning is pretty potent, this method gives us a much stricter limit, 500 milligrams. Uh-oh, the safety margin just got a lot tighter.
And finally, we use the gold standard HBEL method. After that deep toxicological review, the limit comes out even lower, just 200 milligrams. That is our most restrictive, and because of that, our safest number.
So the final decision becomes incredibly clear. You choose the lowest number, because the lowest number provides the absolute highest level of patient protection. In our example, the official MACO is 200 milligrams. End of story.
Now that 200 milligram number is awesome, but that's the total amount of residue allowed across a massive, multi-ton batch of the next product. How you actually measure that on a physical piece of equipment? We have to turn this theoretical number into a real-world test.
Well, it's a pretty logical three-step process. Step 1, you've got your MACO, or 200 milligrams. Step 2, you calculate the total surface area of every single pipe, tank, valve, and gasket that the drug touches. Then finally, step 3, you basically just divide that total MACO across that total surface area. And that gives you a target limit, something like micrograms per square centimeter, that a technician can actually go out and test for with a swab or a rinse sample.
And this all brings us to one final thought. This whole evolution, from a simple rule of thumb like 10 ppm to a deeply complex scientific approach like HBO, it's more than just a change in process. It's a fundamental shift in philosophy. It's a commitment to finding the most scientifically sound limit no matter how hard it is. And it kind of makes you wonder, right? As our science and our knowledge get better and better, are the limits we think are super safe today going to get even stricter tomorrow, all in the endless pursuit of perfect patient safety?